Bismillahirrahmanirrahim. Assalamu alaikum. Ninth class, I am with you, and today's topic is Meiosis I. This is chapter number 5 and cell cycle. Its name is cell cycle. और आज का हमारा SLO है the study of various stages of Meiosis I. So, let's start with our lecture. अच्छा. Now, table of content में हम लोग study करेंगे आज introduction to meiosis, then haploid and diploid cells, homologous chromosomes, and finally the meiosis I. So, आज के lecture के बाद you will be able to know what is meiosis and what kind of a cell can go through the meiosis means के कौन से वो cells हैं जिनमें meiosis possible होती है and what is the difference between haploid and diploid cells and what are the homologous chromosomes, synapsis, and crossing over. These are the major terms of today's lesson. And at the end, I'm hoping that you must know all of these terms. Now, let's start with the introduction of meiosis. So, the word meiosis means reduction division. Please note it down on your books that the word meiosis means reduction division. It means something has to reduce. And in a cell, what a cell can have that can be reduced, it is the chromosome number. So meiosis is helpful in reducing the chromosome number to half. अच्छा ये भी मजे की बात है कि ऐसी बात नहीं है कि पता नहीं कितने कम करने exactly half कम करने like for example if human being has 46 chromosomes then the half of it is 23, right? So meiosis will produce gametes and gametes have half the number of the chromosome as that present in the original diploid cells. Now, it is what is the importance of meiosis? Meiosis is important for sexual reproduction. So, sexual reproduction will take place only if meiosis has produced the gametes. So, gamete formation, then fertilization, and then zygote development is the speciality of sexual reproduction and meiosis. So, another, and not another, in fact, the most important function of the meiosis is maintaining the chromosome number. जितना भी आज तक ह्यूमन बींग हज़रत आदम अलैहिस्सलाम से अभी तक जितने भी ह्यूमन बींग्स हैं they have a chromosome number 46. If there is any change, that change is called as abnormality, right? If it is so that's why nature itself has maintained the integrity of the chromosome number. Meiosis is controlled by the nature. That's why the abnormal number of chromosomes is not common in the society. Another thing is producing variations. So you have seen that we all of us are the human beings. And in my class, if there are 40 students, all 40 students are different from each other. This is because of the meiosis. And what is the importance of this variation? It is going to help us in adapting to our ever-changing environment. Okay, this is the introduction to meiosis. Let's start with its the process, how it is going on. So it is starting with the interphase and meiosis 2 has meiosis, by the way, has two rounds of cell division. One is meiosis 1, the other meiosis 2. So before meiosis 1, obviously a cell has to go through the interphase. Now, what is interphase? So do you know what interphase is? It is the a phase in which cell is going to replicate its chromosome number. Like for example, if human being has 46, it would be 92, and it is going to replicate the organelles which are important in replication or cell division, like centrioles. So they are going to double in the phase and interphase. Interphase is very important, by the way, for M phase to run. So this is a diagram of meiosis. No, it's not the meiosis. This is the interphase, showing you the interphase in which the chromosome is going to double its number, and this is the centrosome as well as there, like two centrioles, you can say. So in this diagram, this is the whole of the meiosis one, all of the stages of the meiosis one. This is the first phase and interphase. Right after interphase, this would start, and you can see the doubling of the chromosome. I'll show you it with the magnifying glass. So you can see in this diagram, every chromosome has two chromatids, right? It means they have gone through the interphase or S phase, right? So, and they are connected to each other with the help of, can you name that structure through which they are going to attach? This is centromere. Okay. Centromere से attached होते हैं. Meiosis one में important चीज है, I have told you that it has two rounds. एक cell divided into two cells, right? These two cells further divided. This cell divided into two cells and this cell divided into two cells, right? It means that a single parent will produce four different cells. But the change in that a parent cell is diploid while the daughter cells are haploid. So it takes place in the cells involved in sexual reproduction. Now what is the criteria? The second question tha na, ki what is what kind of a cells through which meiosis can take place. So this is said the sexually reproducing organism. Diploid cells are responsible. That diploid cell must have a homologous chromosome number and non-sister chromatids. Only then meiosis can take place in a cell. So in this diagram, you can see it is a diploid cell. It is divided through meiosis I and giving rise to two haploid cells. These haploid cells going a simple division called mitosis and producing four haploid cells, which are similar to that, right? Which are similar in chromosome number with that, but all of the characteristics are different from these cells. Now cells which have two sets of chromosomes. अच्छा, अब हमने देखना है कि हैप्लॉइड और डिप्लॉइड सेल्स क्या होते हैं. तो वो सेल्स के अंदर टू सेट्स ऑफ क्रोमोसोम होगा, जैसे कि मैंने बताया 46 and 23. So the cell having 46 chromosome is called as diploid cells, while the cell having 23 chromosomes are called as the gametes. So diploid cell is the cell having double the number of the chromosome, and haploid cell is the cell having the single half the number of the chromosome. Okay. Now look at the diagram below. This diagram is showing you a haploid male gamete. तमाम तर organisms आपने देखे, हर organism जितने भी यहाँ organism है सब के सब में male और female organisms are there, right? Cats and dogs and rats and elephants and lions and सब में lioness, tiger, tigress, सब organism में male और female है. इस तरह human being में भी मर्द और औरत यानी कि male and female organisms are there. Then what does it means? In case if it is a cell of a human being, so in male gamete there must be 23 chromosomes, and in female gamete there must be what 23 chromosomes. Both the 23 chromosomes will combine and will combine means they will fertilize. When they will fertilize, they will produce a cell called zygote, and zygote is having 46 chromosomes. You can see in this diagram. So this is a diploid cell having two sets of chromosome, one set from each parent. Please note it down. What is a diploid cell? Diploid cell is a cell having double the number of the chromosome, and these double the number of chromosome, how this cell can get from each parent, right? Each haploid parent will contribute it all of the chromosome to the cell forming the zygote. Now haploid and diploid cells को थोड़ा डिटेल में देखें. This is a haploid cell, half the number, and this is a diploid cell. हर सेल का आप देखो डबल, हर क्रोमोसोम को आप देखो डबल हो गया. So that is what we are saying that meiosis is a reduction division. Whenever we call it as a reduction division, what does it means? कि it reduces to half. So इस सेल की ओके जी. सो हमने देख लिया कि हैप्लॉइड डिप्लॉइड सेल क्या होते हैं. फिर अब लेट्स another diagram for the haploid and diploid cell. This is one copy of each chromosome. N number of chromosome is called haploid chromosome, and two copies of each chromosome, 2n number of chromosome. This is a diploid organism, right? We all are diploid organism. We all are sexually reproducing organisms. Okay, that's why we are diploid. But the gametes which are producing in our body, gametes form kahan hote hain? In germline cells. And where are they located? As far as males are concerned, in the testes, they have germline cells, and in testes, they are producing the germline cells or the gametes, which are called as the sperms. As far as females are concerned, though, we and all of the females of all organisms, they have ovaries in their body, and ovaries are having the germline cells, and they will produce eggs, right? So हमारी पूरी body diploid है, but only and only the gametes are produced in testes and ovaries. Only those cells are capable of doing meiosis. Okay. So, there is a definition of haploid cell as well, that they contain one set of chromosomes found in gametes and half the DNA of diploids. Diploids से half होगा. This is very clear indication in this diagram, and you should note all these definition for haploid and diploid cells. Diploid cells contains two sets of chromosomes found in most of the body cells. Suppose sexual reproduction cycle. The diploid cells are responsible for having the the meiosis. So the diploid cells, mitosis can take place in both the cell type of cells, haploid and diploid, while meiosis only taken place in diploid cells. Yeah, यहाँ तक बात समझ आ गई है. Let's start with another term, homologous chromosomes. These are the terms we are going to use in our lecture, right? So homologous chromosomes, what are these? So let's start with this diagram in this diagram. From this diagram, this is a diploid cell, right? For example, it's maybe a cell of human being, but it's a diploid cell. This diploid cell has four chromatids, four chromosomes, right? From four chromosomes means among 46 chromosomes. These are from the 46 chromosomes. We cannot show all 46 chromosomes in a cell, so only four are the representing cell. So this is a diploid cell. As this cell and this cell are of same size, both have the same position of centromere, both have the same set of genes as well. Maybe their composition may be different, but the type of genes would be same. Like if it has the gene of height, this may contain the gene of height. Many must contain the gene of height, and it has the eye color. So this should have the eye color, the gene of eye color. So they have the same size, same position of centromere, same color, same type of genes as well, right? This is a cell in having the homologous chromosome number. This homologous chromosome number, for example, 46 chromosomes are there. In a diploid cell, there are 46 chromosomes and 23 homologous pair of chromosomes are there. 23 homologous pair of chromosomes are there. Are you getting it? That means 23 chromosome from one parent and other 23 chromosome from the other parent, right? These two chromosomes, they will meet and they will form a homologous pair of chromosome. Like for example, in this diagram, this is chromosome number 5, and this is another chromosome number. This is another chromosome from the same organism, and its chromosome number is obviously this should be 5 as well. This is from one parent, this is from the other parent, right? So दोनों ही homologous chromosome हैं, ठीक है? और जब आपने ये सेंट्रोमियर है, फिर आपने जब देखा कि रेप्लिकेशन हो गई, यानी कि एस फेज हो गया, आफ्टर एस फेज इसने अपनी कॉपी बना ली, इसने अपनी कॉपी बना ली. This is a homologous pair of chromosome, and these are the homologous pair of chromosome, but duplicated homologous pair of chromosomes, right? These are called as duplicated homologous pair of chromosome. Like this. This is a duplicated chromosome. This is one chromatid. This is another chromatid. Both of the chromatids are called as sister chromatids. This is the region called centromere, and these are the kinetochore proteins, which are basically the binding proteins through which the spindle fibers are going to attach. एक और एक diagram है हमारे पास for showing you the homologous pair of chromosomes. The paternal and maternal. Paternal means father and maternal means mother, right? Male or female. Orange is showing you the male chromosome, and the dark brown is showing you the female chromosome, right? After application, both of the homologous pair of chromosome will form its duplicated copy, and these are now sister chromatid of paternal chromosome, sister chromatid of maternal chromosome. Both of the chromosomes are homologous pair of chromosome. Both of the chromosomes have the same number, same type of gene, same position of centromere, right? So every cell has homologous pair of chromosome. In this case, this is the chromosome from the male parent, and this is the chromosome from the female parent. Like 23, we have 23 of this type and 23 of this type, and all similar chromosome, they will attach it, they will stay together in meiosis. Okay. Then is homologous chromosome की definition आ गई, that homologous chromosomes are chromosome pairs, one inherited from each parent, that carry the same genes at the same location. And while they contain the same genes, the specific version of those genes may differ between the two chromosomes in the pair. Now, phases of meiosis आ गया हमारे पास. Karyokinesis करने लगे हम. Karyokinesis में prophase I, metaphase I, anaphase I, and telophase I. So we are discussing all these phases separately. Okay, जी. After that, हम डायग्राम थोड़ी बहुत आइडिया हो गया है मियोसिस का। अब हम प्रोफेज वन देख लेंगे। प्रोफेज वन विच इज द लेंथी फेज ऑफ मियोसिस, राइट। सो प्रोफेज में सबसे पहले आपको प्रोफेज का थोड़ा सा तो आइडिया है ना, कि इसमें होता क्या है कि प्रोफेज में सब ये सेंट्रियोल अपनी कॉपी बना चुके होते हैं चूंकि इंटरफेज में, एंड बोथ सेंट्रियोल्स दे विल मूव टू द ऑपोजिट पोल फॉर फॉर्मिंग द स्पिंडल फाइबर्स। स्पिंडल फॉर्म करने के लिए, लाइक माइटोटिक apparatus जिसको हम कहते हैं, वो फॉर्म करने के लिए। And all of the chromosomes, they have two sets of chromosomes. All chromosomes are duplicated. Okay. In this case, you can see that the homologous chromosomes, they are not only attached with each other. Not Don't look in this diagram. Prophase one spindle forms around the chromosomes. Okay, ठीक है. So this is the centriole, this is the spindle fiber, the whole of this, the centrosome is the cytoplasm, plus the centriole is called as a centrosome, and both centrosome, they are moving opposite to the opposite poles. These are the nuclear envelopes, the parts of the nuclear envelopes, and you can see the homologous pair of chromosomes, they will attach to each other. Why they are attached to each other, and what is the meaning of this thing, that some red chromosomes have blue parts and some blue chromosomes have red parts. The final of the prophase one should be like this. Before knowing this, we should go to the this step as well, that centrosome, centromere, spindle fibers, and this is the homologous chromosomes, paired homologous chromosomes. They are very close to each other, and this lengthwise pairing of the chromosome is called as, and we have another diagram as well in this page. This is the same diagram. Yeah, look at this. This is the first step in prophase one. The first step is after interphase, and uh, after the the nuclear membrane, disintegration of the nuclear membrane, the chromosome condensation will take place. After chromosome condensation, both the homologous chromosomes, they will come closer to each other. The homologous chromosomes, in case of meiosis, they will come closer to each other, and they pair up, right? This lengthwise pairing up of the chromosome is called as synapsis, right? And synapses will form tetrad. The word tetrad over here, tetrad means four chromatids, 1, 2, 3, and 4. And it is also called as bivalent, as there are two chromosomes close to each other. So this is also called as bivalent, two chromosomes, and tetrad, four chromatids, right? So then after synapsis, in this diagram, you can see after synapsis, the two non-sister chromatids, they come closer to each other and they will form a chiasmata, a cross-like structure. This is called as crossing over. This process is called as crossing over. वो lengthwise pair up क्यों हुए थे? For the process crossing over. Crossing over में क्या होता है? Exchange of chromosomes, right? Exchange of chromosome होता है. Synapsis में lengthwise pairing up होती है, और chiasmata में जीन टू जीन, पॉइंट टू पॉइंट क्रोमोसोम्स जो होते हैं वो आपस में एक दूसरे के साथ अटैच हो जाते हैं। जब अटैच हो जाते हैं तो क्यास्मेटा फॉर्म करते हैं। लुक एट इन दिस डायग्राम, दिस इज वन वन क्रोमोसोम ऑफ होमोलोगस पेयर ऑफ क्रोमोसोम, दिस इज अनदर क्रोमोसोम ऑफ द होमोलोगस पेयर ऑफ क्रोमोसोम। ये दोनों अब क्या कर रहे हैं? They will try to come closer to each other, which is called as synapses. After synapses, they will go for crossing over, chiasmata formation. This is called chiasmata formation. This cross-like structure is called as chiasmata. And what is the purpose of this chiasmata formation? To exchange the chromosomes, to exchange the genes. And you can see in this, the non-sister chromatids, they will exchange few genes. In this diagram, we are discussing only two genes, B and C. They will exchange. Otherwise, there are millions, there are hundreds of genes present on a chromosome, and all of them will exchange their parts to each other. And there are numerous gene variations, right? So that's why we are all different because of this crossing over and chiasmata formation. Okay. The non-sister chromatids, हमने देख लिया था, the non-sister chromatids of homologous chromosomes join each other at some points along their length. ठीक है ना? They are join each other. Such points are called as chiasmata, and each chiasma is a site for the exchange of some genetic material between chromatids. The exchange of segments of non-sister chromatids during prophase I is called crossing over. This is a very important phase of meiosis. It's a long question as well. It's a short question as well. इसमें से पूछ लेते हैं what is chiasmata, what is crossing over. The most important MCQ is which phase is responsible for crossing over? Yeah, which phase is responsible for producing variations in the gametes is basically prophase I. The Prophase I में it is कि इसके features आप अगर देख लो तो it is the longest phase. Chromosomes begin to appear as thicker fibers, जिसे हम कहते हैं condensation of the chromosomes. After condensation, chromosomes they will line up to pair up each other, which is called a synapsis. And a pairing process of homologous chromosomes is called synapsis. And each pair is called bivalent. That means two chromosomes with their duplicated copies is called as bivalent or four tetrad भी कहते हैं इसे हम. This is the whole process of the प्रोफेज वन. देखो इसमें सबसे पहले अलग अलग है, कॉपी बन गई है, लेकिन अब साइनेप्सिस हो रहा है, क्लोजर आ गए हैं, क्लोज होने के बाद क्रॉसिंग ओवर कैसमेटा हो रही है, एंड आफ्टर दैट यहाँ से भी अटैच्ड है, यहाँ से भी अटैच्ड है, सेगमेंट्स एक्सचेंज हो रही हैं और सेगमेंट एक्सचेंज हो चुकी हैं. दिस इज एंड ऑफ द प्रोफेज वन. At the end of the Prophase I, chromosomes are fully condensed and paired chromosomes repel each other. अब दोनों एक दूसरे को दूर दूर कर रहे होते हैं, लेकिन अभी भी कहीं कहीं chiasmata form हुआ हुआ है, इसलिए they can't fully separated from each other. So, however, they are still attached at chiasmata. Nucleoli and nuclear envelope disappear. Spindle apparatus is formed. This is Prophase I. Now, then is Metaphase I. Bivalents are arranged at the equatorial plate. Metaphase is very simple as that present in the in माइटोसिस, जैसे कि आपने माइटोसिस में देखी थी. इसमें भी ये बिल्कुल सेल के सेंटर में, या कहना चाहिए स्पिंडल फाइबर के सेंटर में, ऑन द इक्वेटर ऑफ द spindle fiber, all chromosomes they arrange themselves, but the important thing is that the chromosomes are in the form of pairs. The whole homologous pair of chromosomes they attach to each other still with the chiasmata, and they are lined up on the equator of the spindle fiber. This is very important about the metaphase. Bivalents are arranged at the equatorial plate. Spindle fibers are attached to the centromere, and one member of homologous pair receives the spindle fiber from one pole and the other from the other opposite pole. वो आपने देख लिया. Then is the anaphase. Anaphase is once again the same. The pulling force is responsible for getting apart to separate the chromosomes from each other. In this case, spindle fibers pull the homologous from the opposite ends. Separated chromosomes of each pair move to the opposite pole. Each pole receives haploid set. Now, the anaphase one is responsible to form haploid cells. Ninth class, please be attentive. Another very important short question, बच्चों को पता ही नहीं होता किस फेज में हेप्लॉइड हुए सेल्स, किस फेज में हुए, वो प्रोफेज लिख देते हैं, मगर प्रोफेज सिर्फ क्रॉसिंग ओवर कराने के लिए इंपॉर्टेंट है. लाइक एनाफेज वन इज इम्पोर्टेंट फॉर द टू फॉर्म द हेप्लॉइडी. हाउ कि इसमें आप देखो सारा ब्लू क्रोमोसोम इस साइड पे चला गया, सारा रेड क्रोमोसोम उस पर चला गया, पूरा होमोलॉगस क्रोमोसोम का पूरा रेड क्रोमोसोम उस साइड पे मूव कर गया और ब्लू वाला इस साइड मूव किया. व्हाट डज इट मीन्स? It means the whole of the information present in the red portion will move towards one pole. The blue portion moves towards the other pole. The information half divide हुई है. That's why it is called as haploid. Haploid cells form होते हैं आपके एनाफेज की वजह से, और वो पूरा bivalent move कर जाता है towards the opposite pole. Homologous chromosomes move to the opposite poles of the cell. एक और डायग्राम थी यहाँ बड़ी अच्छी सी, ये showing you very clearly. This is the position centromere. This is one of the chromatid. This is the crossing over के रिजल्ट में जो बना आप देख रहे हो. ये पहले अभी अभी तक ये सारे दोनों एक साथ bivalent की फॉर्म में they were arranged at the metaphase plate, but now in anaphase they will get separated because of the pulling force or the contraction of the spindle fibers, और ये दोनों opposite pole पर move कर गए. Now the last phase of the karyokinesis is telophase. In telophase, is actually the reverse of the my prophase. आपने mitosis में देखा था. Once at their pole, chromosomes uncoil into chromatin. यहाँ वापस condensation decondensation में convert हो जाती है. You can see the chromatin fiber, and obviously if nuclear envelope will appear, so obviously we are unable to see the chromosomes now, because the cell is not dividing at this time. Chromosomes, we can see chromosomes only in the dividing cells. We can see the chromosomes Only in the dividing cells, decondensation is formed here, or chromosomes will convert into chromatid. Nuclear membrane will develop and centrioles will develop, are formed at the round, and both the cells have haploid number of the chromosomes. And cleavage furrow, you can see the cleavage furrow, which is the indication of the telophase, right? If this cleavage furrow is there, it means that karyokinesis has completed and cell cytoplasm is going to divide. Then the division of cytoplasm is called as cytokinesis. In cytokinesis, which is very close to the telophase, cytokinesis occurs by cleavage in animal cell, and although each daughter cell is haploid and their chromosomes are composed of two chromatids, because of this, जब इसके पास दोनों chromosomes हैं, तो it means an amount of chromatin is same in both the daughter cells, but the amount of information is half in each chromosome, in each cell. Means they have divided the information equally, but not this is not the meaning that equal information wise it is each cell has a very separate information, right? But the amount of chromosome is on amount of chromatin is equal in both the cells. But they all the chromosomes आप देख रहे होगे still are connected at the point of centromere. It means for meiosis II there is no need to have cell interface. ठीक है? They must be separated in meiosis II, और meiosis II में इनको लाजमी separate होना है, otherwise division complete नहीं हो पाएगी. ठीक हो गया जी. Thank you so much. This is the end of the lecture. There is a recap. You identify this phase, can you identify this phase? आप ये डायग्राम अपनी नोटबुक्स पे ड्रॉ करें और इसी तरह इसको लेबल करें, इस वाले सेल को लेबल करें और चेक करें कि क्या आप लोग इसको जवाब दे सकते हैं. फिर थैंक यू सो मच फ्रॉम माय साइड. जैसे कि वो साइंटिस्ट ने बताया था, लाइफ कम्स फ्रॉम प्री एग्जिस्टिंग लाइफ, तो बिल्कुल ऐसी ही बात है. एक लिविंग सेल ही दूसरा लिविंग सेल प्रोड्यूस कर सकता है. सो meiosis रिस्पॉन्सिबल फॉर द कंटिन्यूटी ऑफ अ रेस विद कांस्टेंट क्रोमोसोम नंबर इन अ स्पीशी, एंड meiosis लीड्स टू वेरिएशन. आप देख सकते हैं. ठीक है जी. थैंक यू सो मच फ्रॉम माय साइड. अल्लाह हाफिज.